We again starting our class today, and we'll start our class today from the point where we left the last day. And we had studied that the raw materials required for DNA synthesis is DGTP, DCTP, DATP, DTTP. And in the last day's class also we studied that there are these nucleoside triphosphates, and the triphosphates have this alpha, beta, and gamma phosphoryl groups, and the cleavage occurs between the alpha and the beta phosphoryl group. And who actually brings about the cleavage of this alpha beta group is the 3' OH of the primer. Now, as you can see, this is a primer template junction. The role of the primer is to provide the 3' OH, and the template provides the information as to which base will come in next. So I'm repeating this animation once more. Just check that when the DNTP is coming, it is first forming a bond between A and T. When the bond formation is completed, then only the nucleophilic attack occurs. This is a stepwise process which is done by DNA polymerase. So when I'll be describing the mechanism of action of the enzyme, then you'll realize that this step is first and this step is very crucial, because instead of A, let us suppose if a G nucleotide had entered, then TG do not form bonds. So a bond would not have formed here and the nucleotide would have left. But since the base pairing occurs first, and if the base pairing occurrence is properly ensured, then only the oxygen attacks the alpha beta bond of the phosphoryl groups. So keep this in mind. During polymerization, the incoming DNTP base pairs first properly, and then the nucleophilic attack occurs. So I'm just repeating this portion once more. So as you can see, there's the DNTP there. And the incoming DNTP is coming. And you'll observe that AT base pairs first, and then the nucleophilic attack is brought about. So this is how the DNA is polymerized. One after another, the DNTPs are added. Mind it, a DNTP can never be added at the 5' end because there is no oxygen present at the 5' end. It is the oxygen that actually attacks the alpha beta bond of the incoming DNTP. So let's see that once more. So as you see in this reaction, the pyrophosphates were left out. The pyrophosphates are broken down. As a result of breaking down of the two pyrophosphates, the amount of energy released here is used to catalyze the reaction further so that the next DNTP can be brought into. So let's see the reaction once more. G perspires with C, and then oxygen attacks the alpha beta bond, and the gamma beta phosphates leave out from the system as pyrophosphates, which are then further broken to release additional free energy. And one thing is very clear, as you can see, the 3' end has been circled. So unless we get this oxygen available here, the polymerization steps cannot continue. And another thing important is that the orientation of replication, very important, because nucleotides are constantly added at the 3' end. So the direction of replication is always 5' to 3', very important. Nucleotides cannot be added at the 5' end because there is a phosphate present. It is only added to the 3' end. So the growing chain of the DNA shows extreme polarity. That is, the DNA, the chain of the DNA increases in the 3' direction. So as we are seeing that here you saw that in this particular animation just now, they showed that if a wrong nucleotide comes by chance, it cannot pair and automatically it leaves. I'm showing you the repeat of the animation once more. A wrong nucleotide is entering. Since C cannot pair with A, no base pairing, AT base pairing, and then the nucleophilic attack. So the function of the primer or the 3' OH is to help in addition of the incoming nucleotides. It serves as a nucleophile, that is, it attacks the nucleus of the molecule of that incoming DNTP, and the template strand actually guides which base will come in. For example, it was A C had tried to enter, C couldn't enter. T entered, base paired, and the polymerization occurs. The next base is A, again a T will enter. The next base is G, a C will enter. So this is how the process goes on. Now the thing is that this cannot happen automatically. This is not magic. The polymerization of DNA is not magic. It is a very controlled molecular machinery with a lot of molecules playing important role and one after another role in a defined order. So we will now move on to the most important molecule that actually controls the whole phenomena of polymerization. The molecule is nothing but DNA polymerase. It was the scientist Arthur Kornberg and his colleagues who purified and characterized the DNA polymerase of E. coli cells. Later on, there are 3 categories of polymerases that have been identified. DNA Pol 1, this was the first enzyme discovered by Arthur Kornberg and his colleagues in 1955. DNA Pol 2 was discovered later on by Jeffter, Nippers, and Richardson in 1971. And DNA Pol 3 was discovered by Arthur Kornberg's son, Tom Kornberg and Jeffter in 1971. So these are the 3 enzymes basically, which actually plays a very important role in the polymerization process. Mainly polymerase 3, we'll be getting into that, helps in the polymerization process. Polymerase 1 can also polymerize, but polymerase 1 has also another important function. I'll come to that later on. So from this point where we have understood that the DNTP is increased in the 3' direction only, and the primer template junction provides, the template provides the base to be added. Now comes who is adding them, and as I said, the most important enzyme to add to control this whole process is DNA polymerase. So we are now moving on to the enzyme DNA polymerase. At the molecular level, if we just imagine how the enzyme looks like, the DNA polymerase, if you look at here, this is the DNA polymerase. It resembles a hand. It resembles the palm of your right hand. And you can see the DNA substrate. So this is the DNA that is to be polymerized. And it is sitting in a large cleft. You can see the cleft. It is sitting in a large cleft that is a partially closed right hand. And just like in our partially closed right hand, we have a thumb, we have a palm, and we have fingers. Similarly, DNA polymerase, when you draw an analogy to a hand, has 3 domains: thumb, palm, and fingers. It is this enzyme that actually catalyzes the synthesis of new DNA. So as you can see, this is the active site in the palm domain. And this active site is capable of distinguish between RNTP and DNTP. This is very important point. So this is the active site of DNA. And this active site actually is the place where the incoming DNTP will enter. If you look into the structure of RNTP, that is ribonucleoside triphosphate, and DNTP, that is deoxyribonucleoside triphosphate, now observe, for RNTP, there is a 2' OH. For DNTP, oh, there's oxygen here. For DNTP, there is no oxygen. Now, this is the nucleotide binding pocket present in the enzyme. The space provided by this nucleotide binding pocket is very specific. So it allows only the amount of space that is provided, it allows the entry of DNTP because there is no oxygen here, though no extra space is taken. But if by mistake, as we know that in the cell there are ribonucleoside triphosphates also, if by mistake a RNTP enters, it will not be able to sit into the pocket because the place provided by this pocket does not ensure the space required by oxygen. Hence, this is how the polymerase first distinguishes whether a DNTP has entered or a RNTP has entered, and given the constraint of space provided here, it also ensures that only a DNTP can enter and base pair. So I'm going over the animation once more, and now you see what I just now told. So this is the active site, and they are now focusing on the nucleotide binding pocket. This is RNTP and this is DNTP. As you can see, first RNTP will come and try to enter, but this excess oxygen, there's no space for this excess oxygen. Automatically, it is released and only a DNTP can come and sit properly into the channel. So polymerase first ensures that during DNA polymerization, by mistake, a ribonucleoside triphosphate is not added. So polymerase ensures that. Once it has ensured that only DNTP are added, then it proceeds to the next step. As I said, the next step will be the removal of hydrogen from the oxygen and the attack of the oxygen on this alpha beta phosphate. There is one thing that I would like to point it out here itself. The moment a proper DNTP sits here and a base pairing has happened between the A and T, as you can see, that happens between the A and the T. The moment the correct base pair has formed, it so happens that secondary hydrogen bonds are formed to the minor groove of the DNA. So once this base pair has formed, what happens in the palm region, secondary hydrogen bonds form with the minor groove of the DNA, which indicates the polymerase that yes, the correct base has been attached. If there was an incorrect base attached here, these secondary hydrogen bonds are not formed. indicating polymerase that no, there is a wrong base that has been inserted. So as I said, correct base pairing is very important. If G sits here, you see the position of the base pair. They are not aligning properly. So the alignment should be very clear. Once this alignment has been formed, once the proper DNTP has bonded to the pocket, the reaction can continue. You people saw that a G entered and the alignment was not wrong, was not proper, and automatically the G had to leave. I'm just showing the animation once more. Correct base pairing is important. See, the alignment is not proper. Only when T comes, the base pair is formed and the alignment is proper. So once the proper DNTP is now bonded into the pocket, the reaction continues further. So now comes the two metal ions, which we are talking about. These two metal ions actually alter the chemical environment or make the chemical environment such that the hydrogen can be removed and even the oxygen is negative and the charges over, and here also you're having some negative charges. The charges are coordinated by the two metal ions. So as I said, once the correct base pair is formed, as you can see, the finger domain moves to enclose the base pair DNA. As the finger domain has moved in, what did you see? The triphosphate has been moved towards the oxygen in a position where now oxygen is now ready to attack the alpha beta bond. So the position has been ensured. Once more, let's go over this. The moment the finger domain closes, you see, so the conformational change occurs, as a result of which the triphosphate is brought near to the divalent metal ions, and the triphosphate is also brought close to the 3' OH. What happens now? Metal ion A helps to deprotonate the 3' OH. So we have our oxygen. So we got our oxyanion. Next, the two divalent metal ions coordinates the negative charges on the pyrophosphate. Apart from that, there are these lysine and arginine residues present in the finger domain, which helps to stabilize the pyrophosphate when the reaction is occurring. And there is also a tyrosine residue present in the finger domain that holds the DNTP in position when this particular reaction is occurring. So let's go over this once more. As you can see, metal ion A and B has coordinated. And now we have lysine and arginine residues. They hold the pyrophosphates in position. And we have the tyrosine residue attached to the finger that holds the DNTP in the position, and oxygen is now attacking. And the polymerization process is complete. So we are now coming to that part where we started from here. So this was the primer template junction. This was the primer or the 3' OH, to which a new DNTP has been added based on the nitrogenous base found here. And now, as you can see, we have the next base, which has now moved on into the active site. One thing must be clear that the finger domain also plays a crucial role. The finger domain associates with the template region in such a manner that only one base, you can see, only one base is found at the active site. The remaining is a 90-degree direct turn. There is only one base found at the active site. So now next comes the function of the thumb domain. As you saw, thumb domain was not directly involved in catalysis reaction. Excuse me. So what does the thumb domain do? It holds the primer template junction in the active sites, and once a new nucleotide has already been added, it just shifts the nucleotide. See, it pushes back the nucleotide from the active site so that a new unpaired base is now available. So the thumb domain pushes the newly formed pair from the active site to this direction. In this direction, my arrow is showing, and it brings a new base, the very next base, to the active site. So this is the function of, basically the function of the thumb domain, and it also reduces the rate of dissociation of the polymerase from the DNA substrate, which means that it doesn't allow the DNA to open up from the polymerase. So we saw that how the polymerization process occurs in a very defined manner, step by step, by the DNA polymerase. Now the question is that if it so happens that in the nucleotide binding groove, a wrong nucleotide has been somehow added, and that has been incorporated into the dsDNA, double-stranded DNA, what happens? Now, as I said, the palm domain has one important role. It ensures that a proper base pair, base is attached to the template DNA. And once that proper base has been attached, it forms hydrogen bonds with the minor group of DNA as shown by the purple. And this is very important, hydrogen bonds only form if nucleotides are correctly base paired. So if the nucleotides have been oriented correctly, then only the hydrogen bonds form. However, if the just remember, keep another thing in mind, these hydrogen bonds are not base specific. Simply based on if the nucleotides are oriented correctly, these hydrogen bonds. But if there is a nucleotide that has not been correctly oriented, what happens? These hydrogen bonds cannot be formed. So the polymerase understands that a wrong base has been inserted into the growing DNA. So automatically, the moment it realizes that a wrong base has been inserted into the DNA, what happens? The rate of reaction of polymerization slows down. It slows its speed. It has realized that a wrong base has been incorporated because no hydrogen bonds has been formed. So what happens? It slows down, it slows its speed. So the rate of polymerization reaction slows down. Let's go over this animation once more. So as you can see, this is the minor groove. Hydrogen bonds are being formed, and hydrogen bonds only form if nucleotides are correctly base paired. Now what happens? If in a rare event, there is a nucleotide mismatch, the rate of DNA replication has slowed. And now, as you can see, there is another site in the polymerase, which is the exonuclease site. So moment the rate of DNA replication is very slow because of the addition of a mismatch base pair, which has distorted the geometry of the substrate. Then the DNA opens up, the polymerase opens up, and the palm domain is now able to make contacts with the minor groove. Since the palm domain is not able to make contacts with the minor groove, the primer template junction opens up from here and is free to move about and interacts with the exonuclease site. Let's go over this animation once more. Fast, slow. This is a slow rate. Now, mismatch base pair DNA replication has become slow as a result, and there's no hydrogen bonds formed also. As a result, the primer template junction opens up and is free to move about and interact with the exonuclease site. So once, as it will be shown in this animation, that the exonuclear site removes the incorrectly base paired nucleotides from the 3' end. You can see this particular picture, they are showing that when there's a mismatched base at the 3' end, the primer template junction binds to the exonuclease site. The moment the mismatched base is removed from the 3' end, the exonuclease site becomes very slow and the primer template junction then moves to the polymerization site and DNA replication continues. So we end our class today here, where we have studied stepwise the function of DNA polymerase and how the thumb, palm, and finger domain brings about DNA polymerization. We did not do the chemical structure of polymerization. So if I summarize today's class, the incoming nucleotide base pairs are available to the template base. The interaction of the fingers of the polymerase close around the base pair DNTP. The change in conformation of the enzyme places the critical catalytic metal ion in a position to catalyze the next phosphodiester bond. Attachment of the base pair nucleotide to the primer leads to the reopening of the finger and the movement of the primer template junction by one base pair. The polymerase is then ready for the next cycle of addition of DNTPs. In case there is a mismatch base pair, we saw that the rate of polymerization slows down, the primer template junction opens up, attaches to the exonuclease site, the mismatch base is removed, the primer template junction again goes back to the polymerization site. So we end our class here today.